Unit 1, Topic 3. Let's talk about some biological molecules. So the variations in the carbon skeleton allows for a huge amount of molecular diversity. Carbon can form large molecules known as these macromolecules. In AP Biology, we talk about four major classes of those. They might look familiar. They're called carbohydrates, proteins, nucleic acids, and then lipids. Carbohydrates, proteins, and nucleic acids, they all form polymers, true polymers. Lipids, on the other hand, they do not really include a true polymer. And they're also, of course, hydrophobic molecules. But polymers are small components of things that will form larger chains of molecules. So it's important to note that along with carbon, nitrogen is also an important element when we're talking about building proteins and nucleic acids. And we can't forget about phosphorus because it's important for building those nucleic acids. And it's part of that component of that phosphate head in lipids. Phosphate head. Okay, so to clarify, polymers are these repeating chains of monomers. So a monomer is the tiniest unit of one of those macromolecules. And if you have lots of monomers stuck together, we get a polymer. And the polymer is a large chain of those monomers that is just really similar, or it's like identically repeating in unit. And all of that stuff is covalently bonded together. Even though each class of polymer is made up of different types of monomers, the chemical mechanism by which cells all make these polymers, called polymerization, or you might hear polymerization, but it's really polymerization. Hold on a second. Polymerization. All right. There you go. There's the word. So this process is facilitated in large part by enzymes. So the reaction that connects a monomer to another monomer or a polymer is a condensation reaction. It's a reaction in which the two molecules are covalently bonded to one another with the loss of a small water molecule. If a water molecule is lost, this is called a dehydration reaction. Carbohydrate and protein polymers are synthesized by dehydration reactions. The hydroxyl group of one monomer will bond with the hydrogen of another monomer, and this H2O will form. And then that H2O is released. So in a basic chemical reaction format, we have A monomer and B monomer. When they come together in a dehydration reaction, they form AB and water, which is released separately. Here we have an illustration of how a dehydration reaction works. I have two monomers, glucose and fructose, and I'm going to release water by taking the hydroxyl group of one and a hydrogen off of another. Those two, when they come together, it's going to form water. We're going to release water. And the bond between this oxygen will occur with the space right there. Those two monomers of carbohydrates will form together, creating a longer chain polymer called sucrose. I should point out that as an AP biology student, you could definitely be asked to illustrate this, draw this, even if you draw the molecule structures like this. Let's see if I can. All right, so try to switch it. Without drawing all of the little functional groups or other groups that are kind of hanging off of it. And you illustrate it in such a fashion, you can still end up getting credit. All right, so in reverse of that, we have hydrolysis, where we're going to break those polymer bonds by adding water. This is called hydrolysis. So polymers are going to be disassembled in two monomers by the process of hydrolysis. It's essentially the reverse, but hydrolysis means water breakage, from the Greek word hydro, which means water, and the word lysis, which means to break. So the bond between the monomers is broken by the adding of that water molecule with the hydrogen from the water attaching to one of those monomers and the hydroxyl group of the water attaching to the other. So a good example of how hydrolysis works within our bodies is the process of digestion. The bulk of organic material in our food is in the form of these large polymers. And that's too much, and they're too large to enter our cells. So in the digestive tract, these various enzymes, proteins, they basically attack these polymers and they speed up the whole hydrolysis process. When they do that, break them apart, they release these monomers into our bloodstream. So they're absorbed into the bloodstream and then they're distributed throughout our body and our cells. Those cells, they can then use, dehydration to basically reassemble that monomer and or make different types of polymers out of those monomers. So with that, we get a chemical reaction of, I have a molecule AB, that's my polymer. When I add water, I'm going to get it into two components. I'm going to break it apart into two components, monomer A and monomer B. And so here we have an illustrative form of hydrolysis where I have sucrose, my polymer. I'm going to add water, and that is going to take one of the hydrogens from water and bond it to this oxygen, creating this hydroxyl group right here. And then the hydroxyl group bonding in the lost component right here. Essentially, they unlink the polymer. All right, I think you get it at this point. All right, let's just say that you're performing an experiment and it involves a hydrolysis reaction, and for some reason you want to determine how much water you're going to use or you're going to make. And so that polymer that you're working with is called amylose. There's 300 monomers of glucose that make up this polymer. And so how many water molecules are you going to need in order to completely hydrolyze, break down, take apart that amylose starch polymer? All right, so if you think about it, if I have a starch and it's attached to another starch, it's attached to another starch, and I want to take it apart, and it's 300 long, for every two monomers that I'm going to break apart right here, I'm going to lose a water. How many of these guys am I going to need? So if you look at it from a smaller perspective, if I have 3 monomers in my polymer and I want to take it apart, how many water molecules am I going to need? And the answer is 2. I'm going to need 2, one here and one here, to completely disassemble a 3 monomer polymer. So if I have 300, how many water molecules am I going to need? 299. So to check our higher level thinking here, see if you can describe the properties of carbon that make it essential to life. Like, can you list some descriptive properties that make carbon like the success story of all living things on Earth? And then look at the periodic table and see if you can find silicon. It's element number 14. What do you notice about its position in comparison to carbon? What do you think that says about silicon? I'll tell you that there's a lot of sci-fi movies out there that will say that there's silicon-based life forms because silicon has the same structure, valence structure as carbon, it's Si, and offers the same electron valence shell structure to offer the variety of things to attach to it, much like carbon. So it's possible to theorize that if life on another planet didn't start off as carbon, if carbon wasn't as readily available as it is on our planet, that we could have, well, not we could have, but they could have possibly used silicon with a very similar electron valence structure to create the diversity of living things that we see that are carbon-based on our planet. Although there's a little breakdown in that theory because silicon it's a metalloid, so it's not a non-metal. It has certain metallic properties. It doesn't fit into the nice like metal or non-metal type of thing. So actually, it does kind of break down a little bit there, but the opportunity on how it can bond is there.